Isabel Castro Acacio Takuinoa, ko Tasajero Temaunga, ko Zulia Teawa, ko Isabel Castro Mengual Takufaya, ko Jose Acacio Villena Takutupuna, e Fangaya Ho o Tehapu Te Patukeha ki Te Rafiti. I live in New Zealand for the last 31 years, and nearly for the whole time I've been involved with Massey University, first as a PhD student and then as a staff member. And today, I want to talk to you about a project that we have here in the north. The project is actually led by Natikuta and Te Patukeha from the Bay of Islands, and we have lots and lots of people that have contributed to the project because they are all whanau. So there is all the hapu iwi that have been contributing to our project. And today, I just want to talk to you about a small part of it. So next one, thank you. This is we had a couple of students, a couple of PhD students that are here. Malin, who is from Sweden, she's finished. And Ange, who is Teroroa, and she's still doing her thesis. And this is the supervisory team with a very strong Maori kind of ink to it. Simon up at the top, he is Natiporo, and Doug family is Tuhoi, and we have Peter, whose family is Pacifica, and the only strangers here are Malin and me, but we've been adopted, and next. Okay, so we labeled this project the Kiwi Whakapapa project because it is about the genetics of kiwi and what can we do with those genetics when we talk about translocations, because that's a question everybody has. And everybody keeps on talking about, oh, we do this because it's good genetics. We mix the birds because this is good genetics. But in the end, when we sat down and talked about it, we didn't know anything about the genetics of kiwi. So we had no idea whether we were doing this right or not. So we started this project with the idea of helping Natikuta Te Patukeha to take decisions about what to do with those birds on the islands and what to do and where to send them or where to get other birds to come into the islands, etc. So that was the initial idea. So the project has 4 different points that are. You can click once, yeah. So the first part of the project was to have a look at what has already been done in terms of genetics, because there is projects that have already taken place. So what do those projects tell us? Then look at hybrids, because people are talking about the Punui birds. Those are hybrids. They're hybrids between Northland birds and birds that come from Little Barrier Island, Taranaki. So what is about these hybrids? Is it good? Is it bad? People told us a few years back that the birds from Punui Island were probably horribly inbred and they didn't need to be used, and there was a clause in the borders of those birds. They were the dirty hybrids. So we wanted to know, well, are they really dirty hybrids? What's going on with them? Then the next thing is we wanted to know how to age these populations, because not only genetics is something that we worry about, but also how old are these birds? Are we sending birds that are really old places and maybe we have a problem with that? Or what is going on these islands? And then finally, we wanted to develop a management plan that it is actually involved with the genetics of the birds. And the first 3 questions we have already answered, so I want to tell you a little bit about them. The last one is this development of the management plan, which is coming up. Hopefully by mid-year next year, this will be done as well. Okay, next. Now, the first thing is about what do we know about genetics already that was there before we even came along. And this is complicated. You can go back and look at it yourselves. But if you look at this map, if you find anything in orange or in that star in red, that means that there has been a lot of samples collected. About 200 samples for the stars and about more than 100 samples for the orange. And you can see that we have taken lots and lots of samples from Northland. So Northland is the area of the country that has had the highest number of samples that have been collected. Next. This is the map of Northland. You've already seen it several times with the kiwi distribution. And one thing that we have found out is that the different populations that we have that have been sampled here, they have different levels of genetic contributions into these individual populations. And so that's what we call a haplotype. But basically, this is work that has been done with a very, very small part of the kiwi genome that comes from the mother. So this is only information coming from the females in those populations. And you have here, for example, when you see a circle that is really, really big, means that a lot of samples come from there. When the circles are small, means that there is very few birds that have been sampled in there. And the more colors you have, the more genetically diverse is that population based on this kind of system. So there, Purua is the population that had the greatest genetic diversity, and then, but also had the greatest number of samples. And of course, the more samples you get, the better results you're going to get. And then other populations have less information. But of course, this is, like I said, it only comes from the mother. So in the next slide, I can show you that the other thing we found is that we have a cumulative number of samples on this axis. We have years over here. And here we have all the different types of genetic analysis that we have run. And basically what that is telling us is that we knew very little about genetics in 1988, and we have lots of knowledge in 2018. So the differences in these particular tests increases as we get smarter about how we do the work. And the number of samples that have been used for example, these microsatellites, which became very, very important during the 90s, we have lots of samples that have been run on that one, but very few samples that have been run with the tests that we have today, which are the ones that are probably going to help us the best. And at the moment, our work is done with this sampling called GBS, which is called genotyping by sequencing. And this is like, this allows you to see every single gene in the bird's genome. So it allows you to do a lot of more detailed work and understand better those populations. Next. And because we are Natikuta Patuqueja, we have sampled birds in the area that are important to Natikuta Patuqueja and all their whānau and friends. So we have permission to sample birds in all of the populations that are in black. You've written names in there, but we have only been able to sample the ones that are in red, mostly because there were lots of birds there that we could sample. Some of the other populations were smaller. And of course, we also sample birds in Hauturuotoi, because Hauturuotoi was the source population for Ponui Island, and we wanted to know how those hybrids were faring. We also got birds from Taranaki, from the other source population for Ponui. And then we have also dirty hybrids down south in Pukaha and Rimutaka. And since hybrids is an interesting point that we want to have a look at, then we also collected samples there. Next. Now, the dirty hybrids. So this is the dirty hybrids from Ponui Island. And I have to tell you, Ponui Island, it's a very diverse genetically, even though they said it wasn't, is the highest diversity of genes that we have found in any population so far. Now, if you look in this picture in here, I'll explain to you. This you'll see a lot in the genetic analysis that people do. On one side, we have the original population from Little Barrett Island. On the other side, we have the original population from Northland. And in the middle is all of the birds that we have sampled on Ponui Island. There is 120 of them here. So we think our population is about 2,000 birds. So 120 birds is quite a lot of that population. And each little bar represents one bird. And what you can see in this picture is that all of the yellow represents the genes from Little Barrett Island. All of the blue and red represents the genes from Northland. And each one of those birds has a different proportion of those genes. But you can see that we have birds that are nearly all from the same as the Hauturuotoi, and on the other side we have birds that are nearly all the same as Northland. So basically, for some amazing situation, we don't know, we can explain, for some amazing way, when the birds were placed on Ponui Island, they fell in love with each other, and they didn't care, they just mixed perfectly. They mixed exactly right. That's why we have such a diverse population. There were only 14 birds placed on Ponui Island. 6 of them came from Northland, and 8 of birds came from Hauturu. Next. Now this is so we now have run most of the samples. We still have Hauturuotoi, we have Taranaki, we have Motuoroa Island. We have a few samples that we haven't run, that we're running later on this year. But these are the samples that we ran already. So we have Rakomangamanga, which is the mountain of Natikuta and Te Patuqueja. We have Motuoroa Island, Purerua Peninsula, Trouncen, Ponui Island, and Hauturuotoi. And here, we have the relatedness of the animals that we sampled. So in red, I have a line that shows you if you have 50% of genes shared between each other, that's like mom and dad and the kids, right? So that would be 50% band sharing or 50% relatedness. That would tell you that that's the relationship you have between parents and offspring. If you have it above, then you could have a twin, that shares more genes with you. And these in the middle are all of the samples that we ran. So you can see that the less related means that the more genetic diversity you have and the less relationship you have with other animals in your group. You see that Ponui Island is the one that is the lowest. So that's the better off population, in other words. And then you have Trounsem, which represents the Waipoa population, which was the one that was used for Ponui, and Hauturuotoi are very close behind Ponui. So all of them, and that probably explains to us why Ponui is done so well, because it already comes from populations that are really, really well off. And then up there we have Purarua Peninsula, Moturua Island, and Rakamangamanga, and all of these populations have had heavy bottlenecks for one reason or another, whether they're predators or people induced, etc. And you can see that they all are more related to one another than you would, with Ponui. Now, whether that is bad or not, I don't know. I cannot tell you because we haven't yet measured that, and that is what Ange is doing right now. She's looking at whether we have any problems with the inbreeding in those populations. Next. C. So what the main thing that I want you to take home is, one, that what you see in the population is not necessarily what it is. So we have this glorious picture that Malin did, and she has Ponui is the winner because it's got really great genetic diversity, but we wouldn't expect that because we started that population with 14 birds. And when you look at that, people, all these scientists were like, no, that's a bad population, naughty, don't use it, don't do, blah, 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 blah. And then you have other places like Trounsem where you'll say, yes, that's going to be a good population. And indeed, it is a good population, but it's not as good as Ponui. And then you have other places like Rakamangamanga, which is a mainland situation, as well as Purarua, but they have different histories. And because of that, you probably have less genetic diversity. But we wouldn't expect that, And then, of course, Moturua Island, is incredible. How can we have such similar genetic diversity there as you have in Purarua and Rakamangamanga? We wouldn't expect that. We expected it to be much lower. So what you see is not what you get. Next. Now the other problem that we have at the moment is that not all hybrids are the same. So again, we have the same picture as we had before. Over here is this relatedness. Down here is different families that we collected from Pukaha Mount Bruce and from the population at the Rumatakas. And you can see that most of those dots in here are very high up, means that they are really highly related to one another. So they're quite inbred. And those points in here are kind of in the middle, more similar to Ponui. Ponui has a real, great variation in there, but the only birds that we used for this graph were those birds that we knew were already families. So we know that all of those birds have some degree of inbreeding because they're mom and dad and the babies, okay? Just to compare, because these were families as well, so we also knew that they would have some degree of relatedness, right? But what you can see is that the birds in Pukaha and in the Rumatakas are closer related, that it would be good for them, okay? So, yes, please. When you do the same picture for the Pukaha and Rumataka, you find this picture here. Less birds, of course, than we have for Ponui. So I put Ponui in there to show. But what you can see in there is that, the birds aren't mixing in the same way as they did on Ponui. Now, why is that? We don't know. Is it time since translocation? They've only been there for the last 15 years, I think, for some of those, while Ponui has been there for over 55 or something years, so we don't know. Whether the order of translocation matters, we don't know. Next. So, the take-home message. The number of birds in a population, or whether they are on the mainland or a small island, does not reflect the genetic diversity that you have on that island. So, inbreeding has been present in every one of the populations that we've done at some degree, but Ponui is the less inbred of them all. 2, mixing can be good, like it happened on Ponui. Maybe that's a good thing to do. But the translocation plans that you make, they need to consider those things. You cannot ignore it. But the important thing is that each population needs to be tested. You cannot just say, oh, no, I think that this population is going to be all right. You cannot do that because you don't know what you're doing. And we still don't know when is a good thing to mix, when is not a good thing to mix, how to mix them correctly. For that reason, my advice would be stage your translocations. We have the opportunity to make these translocations really amazing by doing the translocations in ways that can inform us of how this is best done. And some of them might not be as good, but we need to try, we need to learn, because the more we learn, not only for kiwi, but this would be for other species as well. And the Patuqueja and Natikuta hapu have a very good idea, because they work on Matoranga, and I really totally support this idea. They think that we should work with these translocations as in a tomo situation, where you get marriages of convenience, if you wish. So this tomo, when you talk to your other hapu, iwi, and you decide this is what we're doing, we're marrying such and such with such and such, and see what happens in this relationship to maintain the bonds, to maintain the genetic lines that we have, etc. I think this is a really great idea that needs to be explored, and I would like you all to go home and think about this. Okay, and that's it. Thank you.
Thank you, Isabel. We do have time for a couple of questions. That's amazing. Who's got some questions for Isabel? Yes, Graham.
We're talking about translocating hundreds of birds in the near future. How are you going to cope with the data from that? Just going to take small samples?
I don't know. Well, that's the kind of thing that needs to be discussed and spoken about. And, you don't need to sample every single bird in your population. So when Malin started her PhD, my first question is, I need to know how many birds we need. So she went back and she did all of this. She looked at all of the other studies. She measured lots of different things, and she came out with this magical number. If you want to answer very simple questions, you use 8 birds. If you want to answer every possible question, you sample 20 per population, and you estimate that the population is around 500 to 1,000 birds. So I think that if we take 20 birds from each population and we sample them, we're going to be quite safe in being able to say these things. Yeah. And at the moment, well, you're going to get all of those samples are there already. So all you need to do is to be friends with Natikuta and Te Patuqueja, and then they might share that information with you. There they are. Over there are Kaitiaki representatives. They can tell you if they want to have business or not.
Excellent. Time for one more. If there's any last questions for Isabel. Oh, there we go, Mike. Yes?
What sort of number of birds do you need before the breeding numbers become so low, with lack of diversity, that you end up with a bunch of banjo players? Is there like a number?
No.
Yeah.
Yeah. So we do have magical numbers. They say 500 is the magical number for a 500 breeding individuals is the magical number. But that magical number changes in New Zealand quite a lot because our populations are quite small. And a lot of the bad genes have been purged out of populations because the bad genes go away when the animals die. So I cannot tell you, there is no magical number. There is no magical number at all. And as you saw with Ponui Island, we put 14 birds in there. We put 14 birds in Moturua Island, and both of those populations are relatively okay. They are not really inbred. They do have some birds that are inbred, but they are not really terribly inbred. So, yeah.
Excellent. Thank you so much, Isabel. Keep up your amazing research and come back to more of our.
Thank you.